that ethos in many, many startups that are ultimately successful. It's taking a really bold vision and saying, like, I believe and I think we can do it through a bunch of hard work and a bunch of perseverance.
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Welcome to the Nucleate Podcast. My name is Sam Kessel, and on this episode, I speak with Joe Horsman, investor at Madrona, about his career and lessons for entrepreneurs as someone who has made the transition from academic to operator to VC. Joe joined Madrona in 2022. He focuses on sourcing and evaluating new investment opportunities across Madrona's investment themes, with a focus on the intersection of life and computer sciences. He also supports the growth and strategy of current portfolio companies. Joe's a scientist at heart and is particularly interested in working with founders combining AI/ML and high-plex multiomics to uncover unique biology. Before joining Madrona, Joe worked in business development at Roche, focusing on accelerating DNA sequencing. Joe came to Roche through the acquisition of Stratos Genomics, where he led business development, wearing many hats to advance Stratos' single-molecule nanopore sequencing technology. Earlier in his career, Joe worked at NanoString, commercializing multiomic technologies, and was an associate at the W Fund, investing in early-stage startups in Washington. Thank you so much for joining the Nucleate Podcast today, Joe.
Thanks so much, Sam, and thanks so much for having me on.
So let's start from your upbringing. Where are you from, and how did you end up pursuing your PhD in biochemistry at the University of Washington?
So I was born and raised in Seattle. I feel like it's more unusual than it used to be, with the giant tech scene growing up here. But truly passionate about the outside and the amazing things that Seattle brings, but also just the local tech scene and biotech scene. And so left to go to California, go to school, and was definitely drawn back by both Seattle, but also just the potential that I saw in this ecosystem. I think it has everything to be one of the next great places to do really impactful research. I think you've seen that with the rise of like David Baker and all the protein design and the homo cell therapy. And so, came back really into some of that boom. With the PhD, I've always been passionate about science. And, I looked around and I looked at people who had interesting jobs, and like all of them had PhDs. And so it was like, hey, I gotta go do that. I gotta go spend the time in lab. I gotta go pay the dues. And, I showed up and you're like, wow, I didn't know anything coming from undergrad, jumping into a PhD. And so it's like this massive learning experience. And, I don't think there's any other way I would have done it. But maybe I was also a little naive going into it because it was maybe more mechanical, where I was like, hey, I'm going to go and just do what everyone else has done. I had the jobs that I was really passionate about, scientists or people leading BD and biotech. I think it's a great place to be at the cutting edge of doing the hardest things that people do, right? New medicines are really, really hard. And like that attracts me to these problems.
So you touched on something interesting there about the Seattle scene. And I'm curious on how you think about it as it still seems to be relatively emerging compared to, say, a Boston or a San Francisco, which have probably a much greater ecosystem in terms of more mature companies coming out. I assume Seattle is on the more early side right now. You mentioned David Baker, obviously sounds like a lot on the AI/protein design thing. So curious a little more to hear about what's the scene like in Seattle? What are you really strong at? What do you think needs to be built up moving forward?
Yeah. I mean, the Seattle scene, I think is emerging. It's been emerging for a long time. There's been some amazing companies that have been built here. And, I think one of the challenges we've had is that companies have been acquired and then subsequently shut down by the pharma partners. So you can reach back, and Immunex was an amazing biotech company built in Seattle. And, you could write a totally alternate history where Immunex becomes a giant pharma. They could have been Amgen, but they got acquired by Amgen. And, Amgen had a big Seattle site for a while. They wound it down. A lot of the Amgen DNA, Immunex DNA spread back through the Seattle ecosystem. You've seen a couple of great acquisitions last year with Chinook and Alpine Immune. These are massive multi-billion-dollar acquisitions, two of the biggest acquisitions in 2024. But, they're not going to become the next pharma now because they got acquired and, they're being reformed into a new startup. And now we see that again with Seagen, of Seagen being acquired into Pfizer. And I'm really hopeful that we don't see a replay of Immunex Amgen. I really hope that Pfizer stays around and is an anchor of this ecosystem. But it's been emerging for a long time where the science here, I think, is world class. We have the University of Washington is one of the largest recipients of grant funding to do basic science research. The Fred Hutch is one of the world leading cancer institutes. We see amazing talent and companies coming out of that. Juno came out of research from the Fred Hutch. Really, the fundamental cell therapy work is still often being pioneered in Seattle. And you see an ecosystem that I think is poised to kind of make another generation of these. There's, the Gates Foundation and the Allen Institute is leading in both the brain science and cell science side of the world from a fundamental research perspective, but also on the AI side. And I think you touched on the AI. Seattle's probably the best place in the world to bring together deep AI talent with some of these early stage life science insight. You have right across the lake from Seattle is Microsoft, and downtown Seattle is Amazon, and GCP, Google's cloud team is in Seattle. It is a tech juggernaut here. And I think that's an opportunity for us to try to take some of this tech talent and plug it into the life sciences as I believe and our firm believes that this is going to be a big wave over the next 15 to 20 years of everyone becoming more tech enabled. And while I think there's a lot of amazing talent in San Francisco and Boston and San Diego and some of these other hubs that are more front of mind, the tech talent I think is real and more accessible in Seattle than anywhere else. And so that's where, I'm most excited. And I think, we are been emerging I think Immunex was founded in the early 80s. It's been 20, 30 years of an emerging hub. And I think we're going to see some really massive companies built. There's been a rich history of that here in Seattle, and I don't think it's going to stop.
It sounds like it's really poised for growth, especially as you mentioned, with the new tech-enabled companies that are coming out there with AI-enabled biotech, which is well positioned. Looking forward, there's a lot of potential with the talent and the relevant tech companies that you mentioned to have a cross-pollination of ideas, especially if I assume you'll have some AI specialist expertise coming out of Amazon or Microsoft that could lend to an unfair advantage for a biotech company.
Yep. And hopefully we can convince them that this trying to solve drug discovery problems, trying to cure diseases is maybe more meaningful than working at Facebook or Meta. I don't want to belittle any of the amazing work these tech companies do, but I make the pitch of you can do massively impactful work and cure diseases with the exact same skills you're using in your tech. And I really think we have the opportunity to draw those people over. Sometimes we can't match the compensation of an open AI, but hopefully there's some meaningful work that drives a great feeling at the end of the day that you are helping humanity in massive broad strokes.
And I think a lot of people are looking for that meaning in their work. So I think it is a strong pitch. So going back to you and your career, talking about how you did your PhD, you ultimately decided to pivot to industry. I'm curious if your experience at the W Fund, which I assume was part of University of Washington, was part of that, or if you found out relatively early on that you were going to make that pivot to industry. Just curious how that all came together, that you made that leap from academia to industry.
Yeah, I was a couple of years into my grad work that I started getting very interested in the transition to industry. I grew up, I'm the only scientist in my family. Both my parents are kind of more on the traditional business end. And, I saw that as a very interesting way to take the research that's being done and apply it to the real world. That was one of the big drivers for me. And so I think it was probably, really my second year in grad school or so, started becoming very attracted to industry and started exploring the opportunities there. Towards that end, I was looking for anything to differentiate my resume coming out of that. It's kind of a harsh reality, but to industry, the vast majority of PhDs look exactly the same. You come out, you have a handful of papers, hopefully they're prestigious, but you really look very similar to every other one of the tens of thousands of PhDs graduating every year. And so I was looking for things to differentiate my resume, show that I had some kind of inkling of being able to speak the language and have an additional skill set that could be valuable from day one. And so that's how I got connected with the W Fund. It wasn't the only thing I did. I had a whole bunch of other things of just experimenting, figuring out things that both I thought would be interesting, but also trying to explore like what I would enjoy doing. As you mentioned, the W Fund was a university associated early stage venture fund. It wasn't, a part of the university, but it was, closely looking to invest in early stage, really spin out companies from the Seattle ecosystem. So looking at Fred Hutch and at the University of Washington. And I stayed there for 2 years. It was so much fun. It was a ton of work because I was, essentially working half or a third of the time at a minimum at this venture fund, but also trying to do my PhD at the same time. And so it was, set up experiments in the morning, go work for half a day, go back to the lab, take the experiments out, then go back to the venture fund, do more work there, go back to the lab in the evening. But it was, it's fun, so I didn't really mind, a little burning the candle at both ends. And it was really amazing for 2 years. And at the end of that, I was kind of like, well, there's no jobs in venture, so that was fun. And it gave me some differential on the resume. Let's go to biotech, right? I didn't really look for jobs in venture coming out of my W Fund experience and after my PhD.
Was that just because the job market at the time was challenging in VC?
I don't even know if I really thought there was even a career in venture. Like I had lots of fun. I wasn't in the Bay Area. I wasn't in Boston. There's very little exposure to like venture capital at scale, especially in the life sciences. And maybe I sent a resume or two in and never heard anything back from, a Flagship or a Third Rock, one of the like blue chip venture creation firms. But really, I think there's also a focus of how can I be a VC? How could I go into this investing world if I don't even know like what biotech is? And so I think my first focus was like, I'm going to go get into biotech and learn the industry. And, I think as almost every fresh PhD learns, like that's a hard jump. Like you don't, even with some extracurriculars and things I've done, like it was still challenging to go find my first gig in industry. And, you work every angle and you hustle because it's, while it's the path, it's, definitely not a given that you can go find your dream job from day one.
Yeah, that makes a lot of sense. And so let's talk about it. Your experience in industry right out of school. You ended up spending a few years at Stratos Genomics, which was ultimately acquired by Roche. So curious what that was like, and sounds like it gave you some deep appreciation of what it's like to work at an early stage company, and in that case, go through an exit and what that process was like. So curious to hear more about that experience.
Yeah. So my first job actually out of PhD was at NanoString. I started off in a product role, product management. And I feel like roles like that, where you can kind of sit 1 foot in lab, 1 foot in the expertise of a deep scientific how to solve problems. I got to think about how a tool or a drug or something fits into the deep expertise you got in your PhD. Like I had to get some kind of validation, and it was really helpful to start off in a role that I kind of could play in both worlds. Go talk to the scientists, but also go talk to customers and lawyers and people who are kind of more on the business side, playing that translational role. From that role, I pivoted to a much earlier stage company, Stratos Genomics. And you're just really lucky if they raised some capital and connected with them and was like, your technology is really amazing. We'd love to chat to see if we can work together. And I think it's many little lucky pivots in your career. Like for whatever reason, I connected with the CEO. He liked the resume. He thought I could hustle. And I think we shared a vision for what could be, and he certainly indexes for hustle, and got hired to kind of do a lot of things that weren't in the lab for Stratos. So while my title was technically business development, I wore a ton of hats. And so I was gonna make the website and fix the website and modernize it, and gonna go out and think about market sizing and how is the best way to approach different applications of NGS with the Stratos Genomics technology. And it was very early. And so there's just these hints and glimmers that like, wow, this could be really amazing if we can keep progressing, we can keep advancing. And it was really fun. I think that's part of the thing about startups. We were, I think, 40 or 50 people when I joined. Everyone was in the lab. Everyone was working really hard. Like when I joined, it was all day, every day. We were like 5, 7, 6 days a week in lab, and like 8 a.m. to 7 p.m. you were cranking and grinding because it was like it's hard, but you're also like in the foxhole with all these people, and you build some great bonds, and you're trying to do something really hard with a very dedicated crew. And so there's that ethos in many, many startups that are ultimately successful. It's taking a really bold vision and saying like, I believe and I think we can do it through a bunch of hard work and a bunch of perseverance. And ultimately, I really enjoyed that. I'm glad I'm not working, consistently like 8 a.m. to 7 p.m. every day. But that's what it takes a lot of times, especially when you're at these critical moments of startups of, when things are working, when things are really at critical inflection points, like you really have to be passionate about the startup and push hard. And we did that. And I was very fortunate, again, like these lucky little turns, like the technology worked. You could make another case where it's like, maybe the Stratos doesn't quite work as well, or Roche doesn't get interested, but the technology works really well. And we were able to sell it to Roche in the middle of the pandemic, and that was a really amazing experience, especially to be sitting in kind of the business seat because you have this front row ticket or front row seat to a really interesting transaction. And the time you're like, let's work really hard because it's make or break for the company. Let's link up with Roche that can bring a scale and an expertise and a sales force that would be really hard to find and build in a startup-like setting. And so it was this, I think, fortuitous and really powerful opportunity for us to partner with Roche and ultimately be acquired by Roche, because it's really hard to commercialize a new NGS tech. And I think Roche now with the Stratos chemistry is in a really interesting position to try to really disrupt the state of play in NGS, which has been a staple for life science research for the better part of 30 years now.
Yeah, so curious a little bit more about the technology, diving in a little bit and how you think the impact of it can reach throughout the industry, especially at Roche. And you wrote a whole article in the Timmerman report about this and about how the recent developments, including time after you've gone, has been pretty substantial in terms of the technology. You highlighted in the article that sequence a human genome in 20 minutes and can achieve greater than 99% accuracy on certain measures. So yeah, I would love to learn more about the technology itself. And it sounds like you wore a lot of hats and had experiences across both some of the scientific side of it as well as the business side of it. So curious at how that all intersected for you.
Yeah, absolutely. So going to click deeper on the Stratos chemistry, it's a really elegant solution, but it took like 18 years to do, which I just think speaks to the ambition and the difficulty of what Mark and the Stratos team set out to build. So it's a short read, high accuracy nanopore sequencing. The fundamental problem and challenge of sequencing DNA with nanopores is that this chemical difference between each individual DNA base is really small. And so you actually are really challenged. It takes real data science and lots of technology. I took Oxford Nanopore years to figure out how to translate little complex signals into true sequence. And even then, it's not really that accurate. So the Stratos insight was, can we take that DNA, which is really hard to differentiate with nanopore sequencing, and copy it into a higher signal to noise reporter molecule? And they do that with a polymerase. So they took a polymerase, highly mutated it, did a bunch of protein engineering that it incorporates these looped nucleotides that have a signal to noise reporter on the end of them. And so you basically copy the direct DNA sequence into its cognate reporter molecule, just base by base by base with a polymerase. And once you've copied your sequence, you chemically treat the sample. These looped nucleotides expand, hence sequencing by expansion, what they call it, and they expand out to 50x the length of the original nucleotides. And then you can just take this long molecule and now pass it through a nanopore, very similar to how people have done nanopore sequencing for a long time. But instead of trying to find these really small signal differences, like, oh, what's the difference between a or a G? Like, I don't know. Now this thing is clear. You can look at the signal by eye and like, oh, I know exactly what that is because the signal difference is so much different. And that kind of fundamental chemistry now makes everything more scalable. Roche can do this in a massively more parallel way. I think Roche is up to an 8 million nanopore chip now. That's why you can get these massive data throughputs. They've solved the problem through really kind of amazing chemistry for the translocation control. So every time you go by a base, because it just ticks by base by base by base. There's no signal deconvolution. It's a really amazing chemistry. I'm actually kind of shocked it works as well as it does, even having worked there. It's one of those grand, ambitious science fiction science projects that I still think people are blown away that it works, but also how well it works, that it works really amazingly well. And I think we're going to see some great diagnostic applications for that. And so I was really fortuitous to link up with Luke and write the article. I think it was fun to give an inside look of kind of like what it took. And no one knew Stratos Genomics was on no one's radar for coming and disrupting the state of play. But now with Roche, I think they are probably the favorite to become the default diagnostic sequencer at this point. Roche is one of the biggest diagnostic companies in the world. And they have worldwide distribution. They have a regulatory group that can rival anyone. And so now I think you're looking at a sequencing world where you really probably have a dominant or a potential for a dominant player to come in and just really democratize and make diagnostics DNA sequencing not only kind of more accessible, but also make it such that you can do more tests, right? One of the key things of the Stratos chemistry is that it's really flexible. You don't have to load your sample onto a giant Illumina instrument and wait 48 hours to get the most cost-effective run. Now you can enter a new regime where it's like, I would like to run a test, and you can do a sample prep, and that afternoon you can get the answer. And it's going to be diagnostic quality. It's going to be integrated into a end-to-end workflow. Like, I think that's going to be a challenge for Illumina because more and more of their growing revenue was this clinical focused, clinical and diagnostic focused DNA sequencing. And now they have a real rival, right? It was not going to be Oxford Nanopore that was going to take the grand prize of diagnostic DNA sequencing. And so, I don't think this is like by far from the death of Illumina, right? They have an amazing platform. They are the gold standard still. But I'm looking forward, like their growth vectors and their growth axes are much more limited with, not only Stratos coming out and the Roche coming out, but, Element is coming up from the bottom doing a like more academic focused sequencer, being very cost competitive. Ultima Genomics is coming out and having very, very cheap reads. I think they're down below $80 whole genome sequence. And so I feel like Illumina is getting a little boxed in. I wouldn't write them out, but it's something I'm paying attention to a lot because I think it's going to be a very interesting few years because for the first time in a long time, Illumina has challengers to the NGS throne and, break out your popcorn. I think it's going to be interesting.
Very curious to see how that all plays out. And, it's come up on a few podcasts that when you have competition, you do speed the acceleration of innovation because everyone's trying to find a way to get a leg up on each other. So I want to ask a little bit more about the implications here as far as having faster, more efficient sequencing. You talked about 20 minutes for an actual doing the sequencing itself. There's obviously a sample prep involved, like you highlighted, but still could be done with the end of the day, is what you're saying. So does this mean then that this opens the doors to more next generation sequencing tests to be used for different cancers, the ability to tailor treatments more accurately, or even derive new insights into what potential mutations could drive a specific cancer. What are the implications clinically and ultimately for people in terms of, are more disease going to be able to be diagnosed more quickly? How do you see this having an effect, let's call it a few years down the road?
Yeah. So I think there's a couple. I'll take one little divergence here because it is a new chemistry, right? This is no longer sequencing by synthesis. And so I think there's like a clinical question, and I'll come back to that. But I also think there's like an R&D question of, I'm very curious to see what like world-leading researchers do with this. Like there's only so much that you can do inside of a Stratos or a Roche of ideating of, hey, maybe this application would be amazing. Their read lengths are actually surprisingly interesting in length. So now they're showing out, and I think this is, incredible. It wasn't this way when I left, but they're now up to like 1,200 base pair reads. Like that's enough to cover a lot of, full-length mRNA, something that data is normally lost with a small read or short read sequencer. And so I'm really curious to see when this goes out in the hands of, world-leading researchers, like here at the University of Washington at the Genome Sciences Building or the Broad. Like, what are people going to do with this? Because I think there is just a creativity now that I'm very excited to see what applications and what very cool like research questions now can be answered, because there's another axis, and it's like I'm not going to be creative enough to figure out all the ways that people start applying this. So that's where I'm kind of most excited. On the diagnostic end, I think the progress will be maybe slower than the research end. I think that's always the case. I think there's going to be workflow differences that are going to be the first ones to hit. It's just going to be easier and more cost effective for many of the NGS diagnostic providers to answer questions in a more modular way. You don't have to wait to fill up a whole sequencer run. And so I think you're going to see faster turnarounds, better workflows. You're more likely to go into a clinic and get a massive genome panel done for a potential cancer or something like that and get the answer more quickly. And that I think will be impactful. People being able to have tailored therapies more quickly. You don't have to wait for a long turnaround time. You don't have to send the sample to a giant lab somewhere, wait for mail in both ways. I think those are kind of the implications that I'm going to look at too first. But I also think that the scalability of this will open up the opportunity to start maybe doing more like smaller panels and having people say, hey, I want to address more targeted questions. If I don't have to quite worry about as much of the like loading up an entire run, if I can just do them one off and give someone an answer really fast, I think there's some interesting implications. Maybe it's infectious disease. Maybe it is profiling for individual metabolism for certain drugs, like the precision like pharmacology. I think that it opens up some possibilities that say, hey, like if we can do this faster and not have to wait for a long turnaround time, we can probably do a little bit more personal tailoring. But that's going to be a slower process because you have to get the FDA on board. You're going to have to really take the time to do the clinical trials to show this is impactful. And so I'm excited for the long run. I think this is a long play. Like we're not going backwards, making medicine less personalized. Over the course of 15, 20, 30 years, we're going to see more and more of these tests spun up. And I think, Roche is well suited to become an engine behind a lot of that. And, to Roche's credit, they take incredibly long-term views of this. And I think they see the world like that there's going to be an increased democratization of sequencing. There's going to be increased use of sequencing across a lot of diagnostics. And, there has to be a sequencer underlying them. And so I think they're seeing that growth over the long time. And that's what I think is getting not only Roche, but other people very excited because we're not using less sequencing. I don't think we've ever used less sequencing year over year. So, it's definitely one of those long science stories where, the cheaper sequencing gets, the more you're going to use, the more applications you're going to find. And this is just the next step of driving that.
And it's really exciting too, to hear across both the research side, near-term clinical, and like you're saying, and long-term seeing some potential innovation in terms of how patients are treated with more precision and personalized medicines. Although you said that will take a while to develop, that will require probably new companies and
Yes.
FDA trials, et cetera. So maybe a longer road, but really exciting to see how that all plays out.
I'm super excited.
Yeah. So You also spent some time at Roche after the acquisition of Stratos. So curious to hear about what that was like going from a small, innovative, nimble biotech where you're wearing 10 hats, where I assume the culture changed significantly once it got acquired. So curious to hear about that. And I'm assuming that has some insights that you were able to leverage for how you act now as an investor at Madrona.
Yeah, I mean, it's certainly different, right? Like going from 50, 60 people to, I think Roche is at like 90,000. It's a different game. You know, I like to think that the culture hasn't totally changed inside of Stratos. There's still like elements of that scrappy DNA of a startup. And it's always a challenge during an acquisition, after an acquisition. But how do you maintain some of the pieces that really made that startup special? But it certainly changed. And I was at Roche for a couple years, and it was certainly an interesting time. It was during the pandemic. And so there was some certain like odd times I think we all had. I wasn't allowed back in the lab because we had headcount, and like since I wasn't actively running experiments for a while there post acquisition, it's like, well, I can't come in the lab, so I'm stuck at home. Eventually got back in the lab, and I think it was really fun to help with the integration, and we got a new building, like going out of the small cramped lab into a beautiful new Roche building. And helping like play a small piece of that process, and it certainly learned a lot. But I think there was a little bit of me that is like, man, it's so hard now because there's now extra layers of politics. There's a challenge of maybe the vision I see isn't the same vision as someone 6 layers above you at Roche, and how do you convey that? How do you make the connection? And people use this analogy. I think it's been used internally at Roche before. It's like these companies are aircraft carriers. They don't turn on a dime. They aren't nimble. They're steadier. They're like Roche is going to be around for a very long time. It's a big stable company, but it doesn't turn quickly. And so I think over the course of my time there, while I love the team at Stratos, and I still head back every once in a while, and I have great relationships. It's like, man, I think I might want to get back to the early stage. Like, I kind of love that move fast and wear lots of hats and less politics. And so I was, kind of looking to get out. It was like I said, I still love the team there, and so it's like also hard to like leave this tech you've been working on for a long time behind. And the opportunity for Madrona presented itself, and so that was the thing that inspired me to leave. But it certainly is a privilege to work at a place like Roche, because you get to talk to people who have been doing a very different size and scale of science, and hearing the way that people think about markets is certainly different than you think about things in a small startup. And it's always a privilege to go learn from people who see the world differently from you, have sat in different seats than you for a long time. So I certainly think highly of Roche and like the things I learned there. I was fortunate enough to keep working for Mark, my CEO at Stratos, and so there wasn't like a total culture shock of like, oh my God, I'm like flying to Indianapolis. If it wasn't going to move to Indianapolis, so there was, at least I got to stay inside the culture a little bit, which I enjoyed.
Yeah, it's a very interesting dynamic, and it sounds like there were a lot of learnings on how a big structured organization approaches problems, and obviously has, like you mentioned at one point, like a very top-notch regulatory team, and I'm assuming a lot more of those considerations come into play just by the nature of the stage of Stratos at that time. That became more and more important as the company developed. So having that level of expertise across all of those functional areas is very important. Although it sounds like on the flip side, things move slower, and by nature, right? Like a big company, you don't want to be pivoting on a dime. That would be really disruptive to everything that's going on at the company. So definitely understand that complicated balance. One thing, I guess, on the people side of things, I'm hearing across these different transition periods you made is that there was some sort of connection with someone that kind of allowed you to make your pivots. So the one from NanoString to Stratos, and then from once integrated into Roche, from Roche to Madrona. I'm curious on how those kind of played out, how those connections with different people materialized. Was it pure serendipity? Are these people that you've known for a long time? How did that play out?
Yeah, it's funny because there's tons of serendipity, and part of it's looking back, hindsight being 2020, but putting yourself out there. I think networking should be rebranded just work friends. Like, I don't think there's much value in going to one happy hour and shaking someone's hand. They're not going to know you, but just these little connections where, oh, I worked in that lab with someone for a little bit, and so I can walk you through each one. I think they're kind of interesting. Of my ticket into NanoString was I knew a woman from grad school, and she started working there about a year ahead of time, and got connected with her. And you're out there looking and just talking to people. She's like, oh, hey, I think they might be looking for someone. That was my into NanoString. And she didn't hire me per se, but it's always helpful to oh yeah, this guy is smart. I like him. He's not a crazy person. From NanoString to Stratos, I actually reached out cold to Stratos, but there was someone in the lab I rotated in that was already working there. And I think he was leading the protein design team at the time. And so I hopped on a call with him at first. And it was again oh, hey, I remember Joe. He's not a crazy person. You get a little bit of this is a real person, and they can hold a conversation and they're normal. And it's like the beer test. Would you want to go out for a beer with this person? Would this person be great to just have in the office and enjoy them, or are they going to be a pain? The connection with Madrona, going all the way back to the W Fund, one of the Madrona managing directors was on the kind of investment committee of the venture fund. We had an investment committee made of a bunch of long tenured industry professionals, and he was on the tech side. It was the investment committee had diverse skill sets. So he wasn't even a biotech investor, but had some connection to Madrona. And it was funny because I first connected with Madrona, and I know them. They've been around for 30 years, and they're a generalist fund. And I was like, I do biotech. You guys are a tech fund. I don't know if this is going to be a fit. And I was like, no, we should chat. And I guess just a little foot in the door of, hey, this person is around the ecosystem, had a small connection, and it's just those serendipitous things, but it's putting yourself out there and making sure that you do know at least a spectrum of people. And it's funny how those come back and you just have no idea what little connection is going to be the thing that cracks the door open even a little bit for you 10 years down the line.
That's fascinating how that all came together, and just sounds like a couple of them were from labs. So that one, I'm assuming you had deeper relationships, but then the one through the W Fund, just maintaining a good relationship with people as you go along and just try to get to know people as you go. And networking has come up a lot, and like you said, perhaps gets a bad rap. Certainly on LinkedIn, people kind of hate on LinkedIn too. But I would say that there are a lot of benefits to maintaining good relationships with people and getting to know people in different ways and being your authentic self when you do it. I think that's why people feel like there's a pain with networking. It feels artificial. It feels not genuine. And that results in people feeling awkward and it's painful. You talk about the weather or whatever. But if you actually try to get to know people and be yourself and crack a joke, that it goes a long way with getting to know people. And like you've highlighted, you never know how that will alter your life trajectory.
Absolutely. I think, the awkward networking events, one thing I've tried to do more in Seattle is just have more kind of open happy hours. And, you go to one happy hour and you meet someone once and it's just that's not really, have no connection, but you meet them 5 times and suddenly you have at least a small thread of an interaction that it's hey, I could actually email that person and do that. We had a conversation about baseball, or, the tap list at the brewery you're hanging out with. It's just the repeated, I've seen that person around the ecosystem 3 or 4 times. That's your work friends. You might not be the real friends. You might not go hiking on the weekend, but hey, you're in the ecosystem. You saw the person at the coffee shop. You're friendly enough to say hi. Over the course of your career, it's not going to be 3 months. It's not going to be 6 months, but over the course of decades, the people who you kind of see around, suddenly you're oh, I do have a network now. And it is those repeated interactions, even outside of your direct day-to-day, whether it's in your lab or whatnot. And so it's been one of those things I've been trying to build more in Seattle as well, because I think you have places like Boston, you have the coffee shop you go to, like, oh my God, everyone here is talking science. And, that's an easier place to do this kind of scientific networking than many places if you don't have like centers of gravity like Kendall Square or whatnot, where you're able to do a network of whether it's some of like the run groups or some of the biotech run groups, like going to run with a person 4 or 5 times, suddenly like they're in your network, they're your friend.
Nothing like sweating it out with someone and then bonding that way. And I will say, going to, say, Area 4 Coffee in Kendall Square, for those familiar, I remember going there for the first time, sitting down, and like every other conversation, someone was talking about siRNA or the newest cell therapy or gene therapy or AI or what have you. And I think there's a reason why, examples like Kendall Square or similar places have incredible innovation. There's just a great cross-pollination of ideas. There's great talent there. You might overhear your neighbor talking, and then you decide, hey, I'm going to chat with this person because they're clearly in the same field. So I think there's an advantage to that. It sounds like Seattle is certainly growing in that regard, and certainly on the AI side, having deep expertise and lots of cross-pollination there.
Yep. Careful with those NDAs at those coffee shops, right? Like you don't be spilling your target there because someone else is going to hear it.
Absolutely true. So let's talk more about Madrona for those not familiar. Can you speak to the overall vision of the fund? I know it has a relatively broad set of investments that it considers beyond biotech, and curious about your particular focus in the biotech side of things.
Yeah, absolutely. So, I feel super privileged to be at Madrona. It's, a Seattle institution at this point. Madrona is a 30-year-old venture fund. This year's our 30th anniversary. As a firm, we've really had the pleasure of backing some iconic companies on the tech side. Our firm got started right in the seed check into Amazon, like truly one of the best venture bets of all time. That company is like Redfin and Rover that, it's like, I use these all the time. It's amazing that was a Madrona company. Things like Snowflake were a Madrona investment. So I've been super fortunate and just a privilege to work with amazing investors. And it is really a apprenticeship industry. And so like, I don't purport to come in and like, I know how to be a great VC from day one. And so it's just a privilege to be able to work alongside people who've been doing this for 20 plus years. One of our founders is still in the office. 2 of our founders are still in the office almost every day. And so it's certainly a privilege to be sitting in this seat and get to learn from some amazing, amazing venture capitalists. That being said, I didn't mention any biotech companies. And it's a new thesis for us. If you want to think about building a venture firm with longevity, you have to constantly reinvent yourself. The companies that were the best companies in the world and the theses that were the best theses in the world 30 years ago, no longer the case. We're not investing in fiber companies anymore. Like that was all the hotness. Web 1.0 is long dead. And part of this, reinvention is always exploring new theses. And for Madrona, we've turned this, intersections of innovation or tech-enabled biotech, but it's a relatively large commitment by Madrona to invest in this intersection of the wet lab data generation, hopefully proprietary data generation. You have a great assay that you are better than anyone else in finding insight, or at least generating the data that has insights in it. But I think as any scientist can attest, like generating the data is like less than half the battle. You need to then look at that data, find the insight, find the nugget of gold that's hiding in high throughput screens or NGS. You're going to sequence terabytes of data, and there's really probably 1 or 2 insights hiding in there. If you're doing a large screen, like how do you find that? And that's where data science and AI and ML come in. And we're seeing more and more, I think, world-class companies need to integrate these two together. And so we're seeing biotech, and we're front row seat here in Seattle, becoming more and more tech-enabled, that you can't just do all your data analysis in Excel and expect that you're going to be a world-class biotech company anymore. You have to think about, how do I bring scalability to the data systems? How do I make sure that the data we generated 10 years ago is equally accessible and can be merged with data we're generating today to make sure there's a corpus of data that is growing and providing value day after day in a growing way. And so we had a front row seat of this at Madrona. We had database companies that we invested on the tech side, and all of a sudden you're like, hey, who's Illumina and why are they our second biggest customer? Like, I've never heard of this company. And I go, it turns out it's really important in the life sciences. And we saw the emergence of Juno out of the hutch and Adaptive out of the hutch. And these are these really cutting edge companies that are sophisticated. Like, you can't just create a company like that with a single small molecule and race it to the clinic. And so we saw this coming out and we formed this thesis. And my partner Matt McElwain is core to forming this thesis. He was on the board of the hutch. And we think that's the way that the future of biotech is. And so now about a quarter of our or about 20% of our financing, our capital is flowing to biotech. And at our core, we're platform investors. We're looking to invest in world-class founders trying to build amazing platforms to tackle really hard biology problems in a tech-enabled way. I'll be the first one to admit, like, not everything is AI. AI is not a panacea. We're not in a realm of, hey, in silico is going to solve drug discovery. Like, we're done, close up the lab. It's got to be lab enabled. It's got to be lab in the loop. You have to think about a regime where you're going to generate data, you're going to use that to make a hypothesis. Then you have to test that hypothesis in-house, and you're going to know if it's right or wrong. And then you have to close that loop. You have to make another hypothesis, generate more data. And by spinning that flywheel, faster and faster and having, low latency testing, then you're going to be able to get to insights faster, and hopefully the insights will be better than if you're just doing a one-off drug screen. And so that's the kind of company we're looking at and the regime we're looking at. I think we're not alone in this. There's lots of people who are seeing more and more excitement and more innovation in these like tech-enabled biotechs. And it's even just like quality of life improvements that are driving the improvements in day-to-day science. So, for example, one of our companies has a Snowflake Streamlit integration to put the data right at the fingertips of the scientists. So they're designing the best experiment they can that day. And it's not just a med chemist saying, I think I want to do this. Like, why do you think you want to do these changes? Or what is the right experiment to do for the machine learning model? Sometimes you need negative data. All the time you need negative data. You can't just build a machine learning model on all positive data. And so capturing the negative data points, or maybe you need to test something that you're pretty sure is going to be a negative data point, but it's going to make your model better. And so just setting up these systems and becoming more tech-enabled is, I think, a 15 to 20 year journey for biotech. It's like we're hitching our wagon to that. I think directionally it's very right.
And it's a compelling thesis too. I mean, there's a lot of hype around AI, which we can definitely talk about. But when we look at the trajectory of it currently, there's definitely applications across, whether it's at the discovery stage, figuring out valuable drug targets, or whether it's accelerating preclinical drug development with in silico models like you highlighted, or even further along when you're looking at clinical trials, making sure that you have every piece of data on every patient that's in the trial, so that you're able to understand potentially super responders to a drug and try to get better insights from that drug and why it worked in those patients. So, all the things that you highlighted around like having proprietary data sets and having unique AI or even just classic data science models would allow you to have deeper insights that can ultimately result in better drugs for patients and faster development of those drugs for patients.
I think a great call out too, Sam. Like, a couple examples I gave were on the kind of the early discovery end. Like, there is so much to do in the entire drug life cycle. Like, a lot of people do focus on this early discovery. Can I find the best molecule? And like, rightfully so. That's very sexy. We need better molecules at the start. But man, there's a lot to do. And you go talk with people later stage, and like, there's a lot to do in the drug development side. There's a lot to do on the regulatory side. We're super excited about looking for companies trying to play what I always term like the enabling layer here, where why should we not have a co-pilot to help write regulatory docs? There's thousands of regulatory docs that go into every single filing, and it's a language, and we have language models now that work really well. And I think we're going to see more and more of these point solutions come up that say, hey, we can solve this and we can solve that. We can make sure that your trial has the highest chance of success because we can analyze more clinical trial data than any individual human can. And, implementing like a data forward approach, not just at the early discovery side, but all the way through the drug development life cycle, and even through like, hey, this phase 2 trial read out. Do we want to resource the next trial? Like your data science can't stop there. That should be a data-informed decision. By stringing all these things together, like I firmly believe that's the thing that is going to move the needle for us, going from, I don't want to say it's dismal, but it's not great chance of success from discovery all the way through approval. Like if we can triple that, let alone like 10x that success rate, like that changes the math for the entire pharma industry. Like that's the thing that I think excites me, but also largely people who are investing in this regime of like, if we can truly 10x the success rate going from early stage to late stage, it's not gonna be one company doing everything, but over the course of 30 years, like that is not only going to build amazing companies and be great for founders and investors and people who are in this thesis, but also great for patients. I would love more drugs and more personalized drugs and better drugs with less side effects. Like these are all things that I think are going to fall out of this kind of tech-enabled wave that's happening. And that's what really gets me excited. And it's rightfully pointed out, not just the early discovery, it's every single step should be tech-enabled. It should be data-driven. And we need the data sets to do it. Like today, we just don't have the data in many of these cases to truly build an ML model or AI model, or even make the most data-informed decision if you're just going to sit there and do good old data science to say, here's the best trial for this indication with this TPP for this drug. Like we don't have the data to do that today. And so being more intentional, like especially looking at the data, is like how I think we're going to get there.
Yeah, and to the point that you made, I think it's north of $1.5 billion on average per drug, and it takes north of 10 years to go from early discovery through approval, if not 15 years. Mm-hmm. So the point that you're hammering home here, that we can speed that up by significant amounts. We can reduce those costs by significant amounts. That would obviously have a substantial impact on the number of drugs that are developed and approved in a given year. So definitely some substantial implications, like we were talking about, across the value chain from discovery through regulatory filings and approval. So very interesting to hear how that all plays out. And now, one of the things that you've talked about too is how do you separate the hype out of all of that? I think, in a way, I see some analogies to the .com era of the 2000s where there's a lot of hype. And I think we may be in a similar moment now here in 2025. There's a lot of excitement around AI. Zyra is a great example of a $1 billion quote unquote seed round. Hard to call it a seed if it's $1 billion. So there is a lot of big investment dollars coming in, a lot of excitement around the space. Obviously, I think 10, 15 years from now, there's really no question about that potential. I think the question from my perspective is, is the short term going to match the lofty expectations that are being set today? Yep. And you wrote this great article about how the word tech bio needs to go. I honestly hadn't even heard of tech bio as a phrase until like a year or two ago, but it is fairly popular on the West Coast specifically. But your article highlights that computational approaches and even AI has been used, including machine learning as well, as a separate discipline than AI, has been used for a long time in biotech. So want to hear more about how you separate out the hype, how you look for some edge in the AI models or the proprietary data sets that you think are meaningful. So curious to hear how you think about that, and how you, with your expertise, are able to separate out the hype from the real potential applications.
Yeah. Well, I think we're, thoroughly in the regime of we're going to overestimate the impact in, 1 and 2 years. I think that's where a lot of this hype come from. But the corollary on that is that we're going to underestimate the impact in 10 years. And I think we're going to look back and be like, holy crap, I didn't think this was going to be as impactful, it's going to be way more impactful than we were thinking. It might not be in the exact places we think it is. But, it's certainly going to be impactful. I think we're going to underestimate the 10-year. in terms of the hype, like I get it, right? Like there's a ton of pitches that come in, and sometimes the AI moniker feels like slapped on at the last minute because like people say, hey, that's what investors want to hear. They want the AI, and I can understand that inclination. I think there's a couple of ways I think about like separating, hype or wheat from chaff. The first starts with the people. Like, is this a credible team with credible data sets trying to solve a credible problem? There's just problems out there where people say, hey, like I said earlier, been in the lab, we got this covered. Like DNA is code, we're smart coders. And like, if the problem isn't credible, if I don't believe that there's any chance of this coming to pass, having spent a lot of years in lab and have some scars to show from that, like that's some hype there, right? Like you're not taking a reasonable shot. That being said, these things are not trivial to build. And so it's like, is this team one that's going to both be directionally right and have the skill sets to move quickly and with agility in a really fast changing landscape? You look at some, like the best example is going to be the protein models. It was like, this month, I don't know who's the best. Is it AlphaFold 3, but OpenFold's going to come out with their open source model really soon. And, Chai or Latent is going to come out in 3 months. And so, you have to navigate a landscape that's really changing. The sands shift really fast. And same thing happens in the tech world where you want to align yourself where other people's innovation benefits what you're building. And if you see people taking incredibly opinionated stances and say, oh, we're going to build on AlphaFold 3 and like we're going to be so integral to our company that we're never going to rip it out. Like that's probably not the best regime because I don't know, in 2 years, is AlphaFold 5 going to blow us away? Like maybe, but I think there's probably a pretty good chance it's going to be better models coming down the pipeline. I also take a hard look at the data, right? Like that's something that you can own inside of a company. If you're generating proprietary data, if you have a proprietary assay that is providing insight into biology, preferably like hard to address biology, like you're putting yourself in a favorable stream and you can go find AI scientists and people to help develop models to get the most out of that data. And, I think we're going to see more and more like commodification of some of these AI skills. People are like, quote unquote, vibe coding all the time. So it's a theme that is echoing in my ears all the time. Like, is this hype? And, is this the right both team and tech and data to be betting on? Time will tell. Venture bets take a long time. But, we are betting on founders who are world-class. And, that's going to be the really big differentiator, I think, at the end of the day. As an investor, you can only do so much to help steer the path of companies. Oftentimes, the visionary founder who is going to run through brick walls. And when a pivot is needed, go do that. And, even if there is some hype that might be in the vision, like if you're backing amazing people, like they're going to be the ones to navigate this landscape that's changing really fast. And I think that's where we're really looking to partner with. It's maybe not the hardest answer. I rarely am like going deep in the code for someone being like, I don't believe their ML model. Because like at the end of the day, the people that you want to back are going to be way better than you can ever hope to be in one of these AI models. You're probably not the best person as an investor to like really quibble over like the exact architecture they chose for their language model.
Yeah, and knowing where you can push and where the innovation is, right? Like you highlighted in the proprietary data sets or how they're generating the data, and more about like what their results are and whether you think the team is backable is more important rather than jumping into that machine learning model. I'm assuming though you have to have pretty deep understanding of what it's capable of and how it works, if not necessarily going onto line 700 of their code to edit that.
Yeah, and there's also just the effect like prove it, right? Like if you're going to make a bold claim that you can design a molecule that binds to a pocket no one else can, like why should I believe you? Go do it. like most of these, like whether it's antibodies or small molecules, if you're going to make the prediction, the actual cost to generate that molecule is rarely prohibitive. Like we have a great set of CROs. And like I think pharma takes the exact same lens of like, hey, if you truly are making bold claims, go prove it. Go make the molecule. Go pull out the insight that you claim is so easy with your AI model. And that's a little later stage question, when you've had a little capital in, but that's when the stakes raise as well, right? Like raising a $1 or $2 million pool of capital, like you can do that on a napkin and you might not even have all the data, but eventually you got to prove it because like that's what our industry trades on. It trades on data. It trades on doing hard things that no one else can do. If anyone else can do it, you're probably not being that innovative, and someone else will do it and probably move faster than you because like that's what farmers are really good at, moving really quickly against known problems with known molecules.
So let's dive into, when you are separating the hype from what you think the true promise is. Let's talk about some examples of investments that you made that you're proud of. I know you're relatively new as a venture investor in the grand scheme of, fund cycles, but, to date, curious to hear about the ones that you're excited about and what you see as the vision for those.
There is a certain level of like Sophie's choice here. Like I love all my investments. I think they're all doing, incredible work. And that is one of those things about life sciences of where, you don't actually necessarily see the day-to-day tick up in ARR in revenue like you would some of these software. One I spent the day with yesterday is a spin out of David Baker's lab. I'll shout out the hometown here. It's a company called Archon Bio. They are 10 years overnight success, like classic story where they spent 10 years in lab doing really fundamental research to design 3D protein cages. And this had a great blip of success. Some of the original applications for this were seen in vaccines. And so you can use a three-dimensional protein cage, display an antigen, creates a great vaccine. You can fix things in geometries that make it very, very immunogenic. And that has showed some success. One of the companies there is Icosavax. But underlying this like 10 years of building, some of the researchers thought there was more here. And so they worked on a technology they call like antibody cages or ABCs. And instead of scaffolding an antigen, they could scaffold a antibody, display out the binding side of the antibodies. And now it's, different. And they showed some really exciting work. They transitioned out of, their PhD postdocs and into like translational investigator program, which is like amazing to have these transitional roles, and spent like another 4 years building up the data set, quietly, like just doing fantastic science, getting in vivo data. And, then just last year, we ended up funding them, leading their Series A. And it's like, oh, this kind of came out and was like, oh my God, this is amazing. Like, how could you do this? Like, well, it took 10 years and a lot of data. And that was showing some amazing work that by having these antibody cages, you can dramatically alter the biodistribution of your antibodies. And so now they, can only go to the places you want or have favorable distribution properties that you might want for a whole host of applications. But you're also playing with things like avidity. And I think the coolest thing, and the thing that got me really most excited here, is you're also layering on a layer of geometry that, antibodies are relatively fixed in how they are built. Like there's that classic Y shape. And you bind to that, and it's like it's going to have roughly the same effect if you're binding the same region. By adding a layer of geometry, of putting in the structured cage, now you can actually change the geometry of things like receptors or channels and lock it into something that's like an agonist or bias the signaling at a certain level by adding this layer of geometry. And that just seems like such a powerful idea. And so I've been super excited to partner with 2 young founders doing this. And, I think it's one of those, maybe failures of our biotech ecosystem over the past few years of like maybe not backing enough young, hungry founders to make sure there's another generation of like iconic visionary scientists building generational companies. And that's what I'm really excited about for, I think almost all my companies, like vast majority of companies we back are young, hungry entrepreneurs who want to do some big vision. I think it's more of like a East or a West Coast biotech style, the founder-led biotech movement. And, super excited about virtually, everyone in my portfolio. Like it's so exciting to see the data mature. And I'm like excited for every single board meeting because I'm like, oh my God, I can't wait to see like the next data set, the next animal data set roll in. And like we're just starting to see a lot of our first like really critical experiments roll in from many of our portfolio companies. And I'm looking forward to a really impactful 2025 and 2026 from a lot of these. I think, people will be hearing more and more about our portfolio companies because a lot of them are, very quietly executing right now, and you'll be hearing more and more about them.
And I think it's interesting that the emphasis on founder-led biotechs. I think the classic East Coast model is you have a venture creation firm that comes in and finds really interesting academic science, and those academics may be involved with the company, but they'll bring in a set of seasoned hands who are able to leverage their significant experience running biotech companies. And I think there is value in that approach too, right? That someone who's navigated early stage funding or navigated through big pivots can offer some insights. But I think the point that you're hitting on here is that the team that actually helped develop the science, who's very in depth in understanding of how the technology was invented in that depth of only someone who built it in the lab would really understand, has a level of insight when they are in charge of the company that they can think about all the potential applications of the science and how to navigate that more effectively when they are built into the company as leadership. So curious your thoughts on how you think about founder-led biotech being successful.
Yeah. I mean, I don't think many people would argue that there's institutional knowledge in any scientific endeavor. It's, there's lots of trials and tribulations in getting anything from point A to point B. So, if you don't have that founder involved, like I think you lose a lot of like, oh, wait, we did that 3 years ago and it didn't work. Or, oh, wait, I have a scientific intuition. I know I said vibe coding. I think there's like some value in vibe biology. Eventually, like there's some insight that you might not be able to articulate or write down, but like, why do I think I should run that experiment? Like there's institutional knowledge that drives that. But I think there's absolutely all the merit to bringing on people who have been there and done that before. I don't think that very many fresh grad students or postdocs fully understand what it takes to run a drug program all the way to commercial, right? And bringing on people, and if you're going to do a founder-led biotech, I think you absolutely should have a group of advisors around you with experience of having taken drugs into the clinic, having launched drugs, having done your specific disease area in a commercial setting. I think it's interesting to go from an academic to an industry setting. Like sometimes the models aren't even the same. Every academic loves this mouse. You go talk to pharma and they're like, no, we hate the mouse, we do the rat. Like, why? Like, probably good translational reasons. And having that knowledge and just like making sure you're running the right experiments within a biotech and talking to the people who have been there and done that before is absolutely critical. And so, when we are doing this founder-led biotech, we need this support system, people who've been there and done that before and have institutional knowledge and have the experience of having built biotech and having put drugs into the clinic. So I think it's a skill set that you almost need to have as you in the fullness of time. Maybe it's not from day one, but if you look at almost every successful biotech moving into the clinic, like there are people who have been there and done that before in the wings advising, and maybe it's a co-founder, maybe it is your CSO or your CMO or someone who has been there and done that before, is really, really valuable. And I just don't think they need to be the founder. That if you can find really visionary CEOs, like why not enable them? Why not let them give it a shot and try to grow into that role with people advising them and trying to make sure that if there are hard-won lessons, like you don't need to experience that yourself. Like you can learn from the mistakes of others and learn from some scars from people who've been there and done that before.
That's great, and makes a lot of sense that you wouldn't just hand a company, especially you're putting a lot of money into it, alone to like a recent grad student or recent postdoc. It's still a team effort. You're still going to leverage that expertise just in perhaps different ways than perhaps the venture creation model would work on East Coast classically. So very cool to hear that difference. So going back to founder-led biotech and entrepreneurs that are in our audience at Nucleate, curious for those who are pitching truly AI-enabled or machine learning or data science-enabled companies, how would you advise them on crafting a pitch where you'll listen to it and say, that's believable? You might still not invest, but that is most accurately portraying their company in a compelling way. Trying to find that balance of like, what are we actually able to do today? What is our vision, and how are we going to accurately plot that course?
Yeah. Well, I mean, I think, go back earlier, it all starts with the founder, right? Like, if you're going to be out there raising VC, like, people are taking a bet on you. And, it starts with having the right level of passion and drive and, the right intersection of, hey, have this great vision for how the future is going to be, and we're going to will it into existence. That's not going to be necessarily easy, but this is the problem that's keeping me up at night. And this is my life's work to do this really hard thing. And we think that this AI is the right way to do it. And I think that's probably the right pathway, as opposed to like, man, I have a lot of good AI. Let me go find what I can do with this thing. Like, hey, let's start with problems. Find really, really gnarly problems. What's the best way to solve these things? I'm incredibly passionate about those problems. I think there's a lot that kind of like shells out from that of like, hey, you're an early stage company. Like, are you being scrappy and lean and, effective and focused? Early on, focus is your one thing that you really have an advantage on. You can push all of your folks to one very narrow problem. Like, this is the killer application. This is the problem we need to solve. You go back to Roche. Roche has a million problems they're trying to solve. That's where you get leverage. If like, hey, we are focusing on this one thing that is the killer application for the intersection of the problem and the tech and the AI that I've built to solve it, and we are laser focused on this. And there might be a million other things that are attractive and interesting, and like, oh yeah, we could apply this to this cancer, and we could apply it to this fibrotic disease, and like that's great. And I think it's admirable that these things are extensible, but you can't do all those things in a startup. And so you have to winnow it down and be like, and then very tip of the spear, here is the killer application. In this funding environment, like, it's tough out there, and I don't think anyone will argue with that. We've really started pushing people to be very early on, not only finding that like killer application, but think about that as like in a product. I think it does, AI companies or any other companies in the platform very little good to just say, hey, we're building a great platform, and we hope it's going to have a use in 4 years once we've finished our platform. It's a much better thing to come out and say, hey, we're building this platform right now, and we have some hints that this is the killer application. This is the product, and we need to build the platform to push this product forward because it's solving this really, really critical need, and there's a biological hypothesis behind why we're solving the problem this way. But also those lessons that we're going to get and that data we're going to generate in solving that problem and solving that, what probably looks like a drug for most of these therapeutics companies also feeds back and makes the platform better. We're going to have to build an ML model for immunogenicity. We're going to have to build a platform model to understand this exact binding affinity. But that lesson is going to translate to program 2, 3, and 4. But we're still focused, and it's like I'll frame it like a program-driven platform is, I think, a receptive message of like going out there, and it forces you to focus then. That's like the kind of regime and the rust, I think, that's finding traction in, platform investors today because of, the challenging funding environment. And, this might be totally dated. This is, mid 2015, like maybe you listen to this in 5 years and it's like all platforms are go. But right now, like that's, the funding environment, and I think where, vast majority of investors are focused.
Yeah, and you already touched on the next question, which I think is very much top of mind for founders. So we're recording this in April of 2025. Liberation Day wasn't too long ago. Public markets are jittery. And seed stage financing, Series A financings have been challenging for early stage companies, and now even grant funding is getting more challenging as well. I think you already covered a lot of it, I think, through your emphasis on focus and not going after every possible application of your technology early, trying to have a strong sense of what the killer application is. But curious on any other ideas you have for founders to navigate this environment. I know you said that exciting science will always get funded, but I'm sure there's a lot of founders in our audience who feel quite frustrated with the current environment. They've had calls with every VC and just feel like they can't get traction. So for folks in that kind of bucket, how do you advise them to kind of stay the course with their company, but get it to the best possible place to get to an investment?
Yeah. I will say I think the best science always gets funded. Now, the bar for best may vary because there's lots of exciting science that isn't going to get funded for a variety of reasons, whether it is, just the applications that's available to or whatnot. There's amazing antibiotic discovery. It's exciting. It's interesting. For this commercial reasons why it's really hard to fund those things in a venture-like regime. I think in the tough times, I'm actually very, very bullish for the future. You go look at the DNA that's being put into companies right now, the culture that's being instilled in companies. This is where iconic companies get built, where people are scrappy and hungry, and that's the only thing that survives tough times. And once you've kind of set that trajectory of the company, that's not going to change in 5 years. Where it's hey, we're at our core a company that hustles and looks after every way to optimize. And those little things compound time after time. You look at when some of the iconic companies were founded, they're founded in bad times very frequently because it forces constraints, and constraints actually sharpen a startup. You need constraints. You can't just say, hey, here's all the money. That doesn't actually generally make the healthiest startups. So I'm very bullish for the future. And the companies that are being funded today, I think we're going to see some incredibly iconic companies founded in these hard times. So I think that's my message of positivity. In terms of how to tilt the deck in your favor, I would say you need to kind of go against any introversion that you have. We are super fortunate to be in the life sciences where genuinely I think people are really nice and really want to help. It helps that you're hey, I'm trying to cure diseases. People are more willing to give you their time. Go talk to people outside of your circle. Go talk to VCs. Hey, what are you excited? What are you worried about? Go talk to pharma, people who've developed drugs in this field. Get a better understanding and get diverse perspectives. Have people who challenge your thesis. I love doing that when people are totally in love with what one of my companies okay, that's a great conversation, I enjoy it. But when people push back and no, I think you're wrong, I'm I am all ears. I want to hear the contra to where I'm thinking because it pushes me. And so go do that, but don't just talk. Go write your thinking down. Clarify where you are trying to build. I think by writing it down, I mean very intentional. Don't use ChatGPT, right? That's not the writing I'm talking about. Use the writing as a tool to crystallize around the value proposition. Think about the killer experiments. Think about the killer applications. Think about the risks of every individual step. Hey, I love this. Wait, is it going to be immunogenic? Oh, wait, is there something coming down the pipe from another pharma that might just render this entire field moot? Be really, really critical and think about every aspect. Try to take the scientific method just on paper for what you're doing. Try to disprove what you're excited about in writing because it really crystallizes. It doesn't let you kind of get away with mental sloppiness and pretend that you understand it. Write down all these bullet points and then go talk to people and rewrite stuff with new knowledge. The last thing I would say is it's really hard. It's never easy. Right? I think we can look back and oh man, it was easy in 2021. And it's it was never easy. And even if the funding might be easy to come by, the day-to-day building is still really hard. This is literally the hardest thing that we do on Earth. Like rocket science is easy compared to drug development. Like rocket science is governed by Newtonian physics. Like we know the math. Like we don't know the math behind biology. People are trying very hard. Like there's these virtual cell models that, people are trying to apply mathematical models to understand biology, but like we're not there yet. It's like literally the hardest thing. And so, it's never going to be easy. But like if you have problems keeping you up at night, if there's a problem that you're like, I must solve this, like go take the jump and go build the company. Like there's no books or podcasts or like any of the reading that you're like, oh, I gotta go do this or my startup's gonna be a failure. Like nothing will prepare you for going and taking the jump. You're like, I gotta go do this. Like just go do it and jump with both feet. Like it does you no good to kind of spend 10% of your time building your startup. Go take the jump after you've talked to some people and written it down and make sure that's a good idea. But eventually you just got to jump and say, I believe and I'm going to make this happen. And like those are the founders and the entrepreneurs that are really successful, where you have this passion and this vision and like you've jumped with both feet and you're going to make it happen. Like come hell or high water, people put walls in front of you, run through them. Like those are the founders who are really successful in good times and in bad times and hard times when something's easy to come by or hard to come by. Like it is inherently a person-driven industry and a person-driven vision. And like if you're gonna be a successful founder, you have to be that person. You have to be that man or woman and just go will it into existence.
I love all of that advice. So, the thing I was picking up on the most on is the writing. I think that's something that we don't hear very often on advice to founders. Like write down your ideas, really go through them, get them torn apart by other people and try to figure out what the holes are so that you can make the best idea possible. If you can find someone who has an authentically different perspective, you'll be able to have a more successful idea. So that, of the several ideas you highlighted there, I think that was one that I find really interesting.
Yeah. Yeah. And don't use ChatGPT, right? That's cheating.
I think a lot of people are using it today, so I agree. It's definitely something that needs to be done in handwriting, likely, in order to have the best effects. So to close out, do you have any final piece of advice for founders in our audience who are going out there, they're raising, they have a great idea that they're passionate about? What's your advice to them?
Yeah. I mean, I would say stay optimistic, right? Like times are hard and building startups are hard, but it only takes one yes to catalyze a round coming together. And so I would say stay optimistic. Like there are going to be amazing startups built that were founded today. And so like I would keep your head up, keep marching forward, because you got to be optimistic about the future. And like the science that I see coming out every day is incredible. Like we haven't stopped doing science. We haven't stopped driving innovation. And so I'm more excited than ever for the future. So like my advice is stay optimistic and keep hustling out there.
I agree fully. I'm excited for all the trends that we've talked about to play out and curious to see what the future holds and curious to see how your career progresses too, Joe. So thank you so much for joining the Nucleate Podcast.
Thanks so much, Sam. A pleasure.
Really enjoyed having this conversation with Joe and hope you do too.
Thanks for listening to the Nucleate Podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.